We are going to start the webinar. We will be two speakers today to present you what are the secrets behind the chemical analysis. So my colleague Emilie Danel, which just is next to me, she is a laboratory analysis manager, and she held a PhD in polymer science and worked for more than 15 years in Rescoll, Applus Rescoll, in the realization and development of analytical methods. And myself, I'm a technical expert in materials and polymer for medical device application. Hold a diploma in engineers in polymer science, and I'm working in Applus Rescoll for more than 15 years as well. So we together work in Applus Rescoll, which is part of the large group AP Plus that I will shortly introduce in the next slide. So the webinar today will be organized as follows. So firstly, I will very shortly present you who is AP Plus and what are our activities. And then we will start deep in the subject of interest by doing like a short introductions, some tips about the regulatory overview and what are the strategy for evaluation. And we will continue by doing some explanation on chemical analysis, and we will finish by a typical example on a medical device product. So who is AP Plus? AP Plus is a global leader in inspection, testing, and certification, specialist in very lot of materials and application. It is divided in four main divisions: Energy and Industries, Laboratory Division, Automotive, and IDIADA. So Applus Rescoll is part of the Laboratories Division, where we perform characterization, validations, and certification of product for many applications, and in Applus Rescoll, especially for medical device applications since quite a few years now. Applus Rescoll, Applus is located all over the world with sites in America, US, and Canada. In America, we have a special site dedicated to medical device packaging evaluation. We have also facilities in Europe, Spain, where there is a headquarters, in France, in Germany, and most of the medical device activities are performed in Europe. And then we also have facilities in China, where we can also perform testing and evaluation according to the local regulation. The global philosophy of AP Plus and Applus Rescoll is to provide a global offer, to be a one-stop shop companies that can just support the customers right from the beginning, the idea, up to process developments, product validation, chemical characterization, product and materials validation, bring a deep expertise on the characterization we are performing, not only just performing characterization, and then go through the whole life cycle of the product by doing also some certification. Typical activities can be divided to be testing, so on routine, on-demand, customized methods, both for have a deep knowledge on the materials, products, and byproducts generated by the application. So we can do physical, chemical, mechanical, thermal, microscopic and surface analysis, and of course, chemical analysis. And in the frame of the medical application, we can also perform validation, biocompatibility, packaging, transport, stability or cloning. Most of these activities are accredited ISO 17025, and I should say that there are several sites in AP Plus that holds this kind of accreditation and certification to be assured to provide suitable and efficient result for product. So here on the right, you can find just typical services we can provide on routine contamination, material properties, reverse engineering. So if you have a question on your product, you can ask us to understand the product, or go deeper in the product. So now going in details in the subject of the webinar, I would like to do a short introduction to just on medical device word, what are biocompatibility, when should be taken into consideration. So as you might know, a medical device is not only one product and shows a wide variety of products, either implantable, non-implantables, direct or indirect. So what are the definition of a medical devices? A medical device is an instrument, an equipment, a product that is intended to be used as medical purposes. So all these products must undergo a conformity assessment to demonstrate they meet legal requirements and that they are sure, safe, and perform as intended. Risk-based approach is a typical approach and mostly an expected approach when developing a new medical device, with the concept of biological risk, their identification, and the way to address them is today's very important, and is clearly the nest of the development of medical devices. So when we are talking of biocompatibility, this is the general term to verify the ability of a device to be in contact with living organism without causing an unacceptable adverse effect, and that could bring in some unacceptable risk while used during its intended use. So we can wonder when should be taken into account biocompatibility, how it's comply with regulatory requirements. So there are several main steps when talking on the life cycle of product, and especially developments of a new medical device. We start generally with the design, the concept and the definition of the product, then going through the prototyping, the evaluation, validation and the product, and finally the technical documentation. So when developing a new medical device, the evaluation of the biocompatibility and associated risk should come really at the first step, either the concept design, because in this stage, this is the step where we define the intended use, what are the addressed population, what level of risk could be induced by the medical device, and as well as the evaluation of the benefits-risk ratio to promote the use and the commercialization of the product. So considering biocompatibility is not only a requirement at this stage, but it could help in the decision and the selection of design materials to use process, as well as appropriate supplier that are aware of the requirements in medical devices. So once addressed as early as possible, it will really help in the conceptions and the prototyping phase, and then it can be verified during the qualification, and it will be used as input for the technical documentation. So depending of the region of commercializations, the referent authority to demonstrate the safety and performance of the medical device differ. Talking about Europe, legal manufacturer should meet the so-called and known MDR, or medical device regulation 2017-745. So the MDR regulation address a certain number of general requirements for safety and performance and address the management of the medical devices itself, but as well the definition of biocompatibility according to the level of risk. And as a risk category, the medical device, as said, should be safe and performant during all its life cycles. That means, and it should be also safe during the normal condition of use, and as said, should not induce adverse effect on patient, on users. So that means that considering biocompatibility should address its compatibility with environments of the component of the medical device, but the medical device itself, the materials interaction in between the different components, accessories, and part of the medical devices, the properties of the materials at different stage of the use, after, before, storage, and transport to help to anticipate risk of degradation of the product. And as well, it should be taken in consideration compliance to other regulations. So we are talking about a chemical regulation, for example. REACH regulation is an important regulation to follow up during the development, as it could change the risk and the dangers induced by the product. But we have also RHS, PPWR, or many other requirements. So based on this requirement, the biological safety strategy and evaluation can be drawn. Issued from the global regulation requirements, we have as well in parallel some guidelines that address the methodology to approach the biological evaluation. Especially in Europe, we can find this series ISO 10993 that provide both the general methodology as well as giving specific condition of test. Once confirming the product is falling into medical device regulation, the biological evaluation strategy is a process where potential danger, associated risk, and the acceptability criteria can be identified and addressed. It should be planned along the whole process and should ensure that providing strong, adequate, and sufficient data. So the global methodology and the global strategy for biological safety assessments consist firstly to categorize the medical device. So this category is different to the one we can find in the medical device regulation, because here we want to identify if the medical device is in direct or indirect contact, what are the duration of use, what kind of contact, is it in contact with the skin, is it in contact with the blood, the nerves, is it in contact with damaged part of the body, or is it in contact with intact part of the body? So based on this categorization and based on available data, it can be identified which kind of tests are required and how they should be performed. So chemical characterization is one of the pillars of the biological evaluation, because it comes straight after the categorization and before biological test. The physical and chemical characterization can help in understanding the composition and potential release of product. As we have seen, the global approach is a general strategy going step by step, by identification, chemical characterization, biological test, and then reported. But it has to be taken in consideration. This is an iterative process that is based on the building strong and scientific argumentation. Based on the information of the description of product, we can gather available information on composition, physicochemical characterization, information on manufacturing, packaging, sterilization, but also taking into consideration the toxicological risk of single components and raw materials is important to establish the biological evaluation plan. So we can see here, we can just go firstly collecting the data, doing the gap analysis, and then we can address what kind of tests are required to complete the missing data about the material as a medical device, and then do a risk assessment, risk control to make the report. But we can go back and forth according to the development of the product. So very few words, even though it's not the topic of the subject today. There is a new version of the ISO 1093.1, which gives a global methodology. More and more, the global approach should be based on risk management philosophy, which get aligned with the risk management standard ISO 14971, and complete an iterative process between available data and generated data. We should also take in consideration the concept of bioaccumulations in the environments and take in better consideration all life cycle of the medical devices. And finally, the last but not least, there is a strong importance on scientific argumentation and therefore the control of the materials, the product, and the process. So once collected and argumentated data are gathered, it's time to generate missing data to complete the biological evaluation, and it starts with the chemical analysis. And I will leave the words to Emilie.
Thank you. So we will now focus on chemical analysis that we can perform on medical devices, and we will see how it can be impacted by the type of medical device and which kind of chemical analysis we can perform. So as Marilys presented, a medical device is not only described by its chemical nature, but is also affected by several steps during its manufacturing. Based on this information provided by the manufacturer, the laboratories such as Applus Rescoll can determine which kind of analysis will be performed. So we need to know the chemical nature of the medical device. The main medical devices are made of metallic or ceramic or polymer materials, but we can also find some adhesives or ink or also additives. And the medical device is also affected by processing and cleaning agents. So the manufacturer has to gather several information for all these compounds, and it can be helped by a supplier or manufacturer of the products that are used in the manufacturing of the medical device. And the manufacturer can also have a lot of information based on previous study or previous materials developed in the company or research and development studies. And we can also find a lot of information in literature based on previous developments. If the amount of available data is not sufficient for the biological evaluation, the next step is to generate this data by chemical analysis. The first step of analysis when we receive the medical device in the lab is to perform extraction. The extraction step is the first one and also the most important part of the chemical analysis. The objective of this step is to extract compounds from the medical device. Medical device most often is a solid product, and compounds can be on the surface of the medical device, but can also be trapped in the medical device. So we have to use solvents that can be polar solvents, semi-polar solvents, or non-polar solvents, depending on the use of the medical device. We use also the information provided by the manufacturer on the type of contact, the duration of contact, as Marilys presented, to choose the extraction condition, the temperature or the duration of extraction. So all information provided by the manufacturer can be important to determine the best way to perform extraction. And it can include dimensions of the medical device and complete description of the device. The expertise of the laboratory is to choose the best solvent in order to avoid degradation of the medical device, and also to choose the temperature of extraction depending on the chemical nature of polymer medical devices. After the extraction steps, we will proceed to a lot of chemical analysis in order to identify volatile compounds that can be, for example, residual solvents. We can also analyze semi-volatile and non-volatile compounds. All these compounds are organic, and they can be monomers, additives, or small oligomers extracted from the medical device. And we can also analyze ionic compounds or inorganic elements, depending on the cleaning process or nature of the medical device, if it's made of metallic parts. If we go deeper in the description of the chemical analysis, ionic substances are analyzed by ion chromatography. And this technique is a quantitative analysis, meaning that we can determine the exact concentration of the extractables. And we are sure that the elements detected are the one indicated in the reports. In the same way, for inorganic elements, we use induced coupled plasma, and it's also a quantitative analysis based on ICH Q3D regulation. For organic compounds, it's more complicated, and we only have a semi-quantitative analysis. We use gas and liquid chromatography techniques that are coupled with mass spectrometry detection. This analysis requires additional interpretation because we need to interpret the identification proposed by the technique. So we will spend a little more time on these two techniques that are GCMS and LCMS. So first of all, why do we use mass spectrometry detectors for this analysis? Most of chemical laboratories work with more universal detectors, such as GCFID or HPLC UV. These are less sensitive detectors than mass spectrometry, which can impact the result for medical device regulation, because manufacturer has to prove that there are no extractable compounds that can affect the patient, and they have to reach a very small amount of compounds. So we have to use the more sensitive detector available to provide a good interpretation for medical devices manufacturer. HPLC UV is also very largely used in the chemical field, but UV detector is not universal and is very not sensitive. So it's not possible with these detectors to go further in identification of compounds. We need to have and to know the compound that we are looking for. And this is not the case for extractables study. In these studies, we perform screening, and by screening, we mean that we will identify all detected compounds without preconceptions. We need to understand what we see, but we are not looking especially for a list of compounds. The identification of compounds based on this chemical analysis are quite different depending on the technique. For GCMS results, we have a universal library such as NIST library, because this equipment will lead to the same result, whatever the technique used, whatever the instrument, and whatever the laboratory. So all the same compound analyzed everywhere in the world will give the same mass spectrum, and it is then possible to have a database that is very large, because this library has 350,000 compounds. We perform the identification by comparing the mass spectrum obtained in the chromatogram of the medical device extract compared to the library, and see if we have a similar spectrum with one or more compounds. Sometimes it's obvious that we will have the right compound because the mass spectrum is very specific to one compound, and sometimes for families such as hydrocarbons, for example, all compounds of the same family will have the similar mass spectra, and it is more difficult to determine the exact molecule formula or the exact molecule name. And then we can also have information about the family or the presence of some parts, for example, aromatic rings or long aliphatic chain or ester function, for example. For LCMS identification, the work is more difficult to perform because every equipment, every method, and every lab will have different results, and we can't use a universal library. Each lab has to create its own library based on its method and its work. The interpretation is based on exact mass measured by the detector, and calculations are performed to determine the chemical formula that can correspond to the exact mass measured. It is difficult to distinguish between molecules when we have only a chemical formula, because when we check, for example, a chemical formula with 12 carbons, 24 hydrogen, and 1 oxygen, we will find more than 4,000 molecules on PubChem. So it's difficult then to say it's this molecule and not another one with the same formula. This is where the expertise of the laboratory can improve results by interpreting all these molecules to find maybe a more precise assignment. And this can be done by using knowledge on polymer or material science, on medical devices or chemistry, and also all the information that are given by the manufacturer about cleaning processes, manufacturing processes, and formulation. We will now go to the last part of the webinar, which is more information about the product and how we can link chemical analysis results and knowledge of the product to be more precise on the results that are given to the toxicological study. So we will speak about some generalities, and then we will use 2 case studies that we see and give you additional information. I will let Marilys speak about the next slide, if it comes. Yes.
Yeah, so as you have seen and as it was explained by Emilie, sometimes it's quite hard to understand and have a precise and efficient results, chemical results, just by doing the chemical analysis, either by GTMS or LCMS. The interpretation can be really diverse according to the feedback and the knowledge of the laboratories. So just having no information on the product could make the interpretation rather difficult. And at the end of the day, that will come. We will have a non-product that will be a very high level of risk. So in addition to chemical analysis results, what can be done in collaboration in between the legal manufacturers, the developers, and the laboratories is to collaboratively work on getting precise informations. So most of the time, the source of extractables and leachables on the medical devices. So firstly, it can come from chemical composition, formulation, for example, of the product. As Emilie said, we can have specific additive like thixotropic agent, ink, very functional agents that bring specific functionality in the product. But we could have also monomers associating to not complete reticulation of the polymers, which happen quite often with some polymers because it's hard to reach 100% reticulation. And if there are not proper post-treatment after, we can have some left monomers inside the medical device. And most of the time, these monomers have quite low molecular weight, so they have a facility to migrate through the medical device and be extracted from the solvents, and we can find them back in the chemical analysis. So we can have also our source of extractables coming from process additives. So this is not directly the raw material composition of the medical device and not a specific component of the medical device, but it comes from the environment. It can be mineral oil, for example, when talking about machining. Sometimes we need to use mineral oil. We can find as well some demolding agents or processing agents that helps the system to be properly manufactured and fabricate for the final product, especially when we have complex product, complex design, or complex process. This requires quite a lot of use of several additives. Also, it's not directly a specific additives, but we can have also the materials parts in contact with the materials during the manufacturing process. So sometimes we do not always think of this because this is not going into the composition of the process nor the composition, but it could have an impact on the chemical analysis. And we can finally have like also cleaning process using solvent, detergent that is required to make the proper cleanliness or sterility of the product. We could have as well, it's not written here, but the impact of the sterilization and additives coming from the packaging, because sometimes the packaging could be leaked out from the packaging into the medical device that could induce and could be a source of extractables. So when it's intentionally added substances, component, additives, we can have information based on the material safety data sheets, the certificate of analysis, but as well on regulation requirement conformity, like REACH, CLP regulations. We can, beforehand, doing the chemical analysis, identify the potential presence of toxicological toxic component or potentially toxic compounds. We are talking here on CMR, which are strongly regulated by REACH, but as well in the MDR regulation. We can have some substance of concern. The list of substance of concern is daily, not yearly updated. So there are more and more substance that is coming into the substance of concern list. So I have a frequent up-to-date on the evolution of REACH is important because it could induce more risk or more need to make analysis. I think you have heard as well on PFAS, which are the eternal pollutants, lively pollutants that could also have a strong risk of toxicity, but also in bioaccumulation inside the environment and then having a risk for environment and human. Endocrine perturbers, PMB and PBT is very bioaccumulative and very persistent product. That is a new category that is coming to monitor and control the risk of regulation. So you can require from the supplier all this information to have like a strong knowledge on your product and on your manufacturing. But there are also some other risks and other source of extractables. For example, if there are specific degradation between two components that could lead to decomposition, degradation, or bioreaction on the product, especially if the process is not well monitored, you can find some impurity. If the selection of the supplier does not require specific purity, for example, or the contact part of the manufacturing process that could be an origin of source of extractable, and then we have sterilization and final conditioning. So this is where it comes to the strategy of the testing laboratories to have good information.
Yeah, because additionally to the chemical results, the aim of the testing laboratory is to allow the manufacturer to perform the less toxicological study, because this is expensive study, and if we can help to minimize the impact, it's important to go deeper in the results. So that's why the laboratory work is to check the coherence between the medical device composition and all information that Marilys described before, and the chemical nature of the extractables that are detected. Then this is to evaluate the potential source of these extractables by evaluating the process, the formulation, and also degradation mechanisms that can be determined based on the composition. So this is not so easy to determine the exact chemical nature of the extractable compounds each time, but we can perform additional investigation to help. We will now see two examples to give you some additional investigation that we can perform.
Yeah, this is the first example is to show you how we can proceed step by step to increase the knowledge and increase the understanding of the chemical composition of our product. So here we worked with an atopic scream analysis, which is like a very complex formulation. So you can find on the right basic examples of the composition of a cream with general raw materials we can find and what are their main function and why they are needed. You can find there are more than 12 raw materials with thickeners, lubricants, emulsifier, preservative to help the product to be stable over time. Thickener is just to help the cream to stay on the skin, for example. So we performed an exhaustive extraction based on the, as Emilie said, ISO 1093.12 that gives the conditions of the extraction according to the application in order to do semi-volatile, non-volatile, and volatile organic chemical analysis that I'm sure that you'll completely better understand with the explanation given by Emilie earlier. We used the two solvents as it was required and discussed with the customer. There were more than 59 molecules that were extracted from the different solvents. So firstly, we just verified each molecules and see ensure that it was in currence with the expected formulations and global formulation. Silly examples. If we can see biocide, well, for sure, this is not consistent. So we just know that we can eliminate, or we need just to think back on the database obtained by the chemical analysis. So going step by step with the information from the customers on the exact composition of the product, we could find that identification of most molecules. But even so, doing this comparison, there were still 15 molecules not identified by a specific molecule. So these results were unusable for advanced toxicological analysis. And after discussion with the customers, we needed further investigation. So how we did proceed in this case, firstly, we asked the customers to give us a specific reference of the raw materials used, the materials datasheet and the technical datasheet, to see if we can find some additives. We did look on the raw formula and looked at the interaction in between the different components, and we find out that we could find that the presence of triglyceride, which is a typical fatty acid decomposition product, and going back to the formulation, we found the fatty acid. So we could link the molecule with these raw components and give better consistency on the results. And the second step was to discuss with the customers on the manufacturing process, steps, time, temperatures, kind of manufacturing. And we identified a step where they could have like a chemical reaction of one compound. And going back to the discussions, at this stage, there is strong collaboration with the legal manufacturers to get information. We could identify that finally it was not a surprising chemical reaction, as it's well known. So based on that, we could again identify new molecules, and at the end of the day, there were only 4 left an unidentified molecule. The potential to go further is to evaluate the fragmentation molecule and doing specific molecule analysis on raw material. And this is the final example of giving by Emilie.
So the last part is the analysis of specific molecules. If we interpret results and see that we can go deeper in the identification, we can confirm this identification by analyzing the standard molecule and see if the retention time of this standard molecule corresponds to the retention time of the suspected molecule in the extract, and then improve the level of confidence in the identification of the compounds. We can then perform external calibration using this specific standard in order to obtain the true concentration of this molecule, which will help also for the toxicological study, which will last less time. And it's also a better argumentation for notified bodies if the manufacturer has all this information and has the highest level of confidence. The last case study was the analysis of a medical device based on the glycerol. When performing the analysis, we detected a glycidol molecule, which present a toxicological risk. So the next step of this study was to perform kinetic analysis in order to see every 24 hours the rate of extraction of this compound. Then we performed a specific analysis. We analyzed glicidol molecule and confirmed that it was the right molecule detected in the extract. And then we quantify the amount of glicidol really extracted. And we also performed some tests in order to see if glicidol could be created during the analysis by degradation of glycerol when injected in the gas chromatography equipment. And we could see that it was not the case. So finally, we changed the solvent in order to select the most the solvents that were simulating blood contact. Which is ethanol 50%. And we saw that in this solvent, glycidol hydrolyzes and is then not more detected. So the risk for the medical device in the final use of this device could then be neglected. And the manufacturer gathered a lot of information about his medical device to see that if it's made of glycerol and used in different contact condition, it could be problematic due to the formation of a glycidol compound.
So it's time for us to conclude, to make the conclusion of on the webinar. More than conclusions, we have a few things that we wanted to highlight and emphasize when performing chemical analysis. So firstly, chemical analysis are more and more challenged by the regulation and by the evolution of the different regulations. So that illustrate perfectly that clearly we need to go more and more with an exact identification of extractables. We said it several times, but I think it's main sentiment. It's really a teamwork between the manufacturers and laboratories, and it's a key to have success in performing deep, current, and strong chemical analysis for both toxicological risk, but for better understanding of the medical devices. So the more you anticipate the formulation in the device conception and design, the more you can minimize the risk, work on improving the biocompatibility, and not facing big default and big risk of biological risk that could be identified at the end of the qualifications. That would mean that you will need to get back to better understand what's come from the risk. We say it again, the more the device is known, raw materials, additives, manufacturing process, the more precise will be the chemical results, and the more efficient and coherent will be the results, and you will be able as the to rely on the conclusion of the chemical analysis. And last word, keep in mind that extractables that are not only intentionally added substance, but can be non-intentionally added substance, as has been shown in our different examples. So this is the end of the presentation. We have 10 minutes left for questions.